that plants can compensate for rubisco being so sloppy and taking in either carbon dioxide or oxygen. And so we were looking at the C4 cycle before we had the exam on Friday. Oh, so the multiple choice portion, everyone's done taking it now. So I'll reserve time here and we'll go into that toward the end of the hour, okay, and look at the results. All right, so we've been looking at how plants have evolved mechanisms to try to deal with rubisco being so carefree about taking in CO2 or O2. So we looked at C4 plants and this idea that basically when you look at what they're trying to accomplish, it's this idea of trying to get CO2 really concentrated around rubisco. So that rubisco will only take carbon dioxide into its active site and ignore oxygen because the CO2 will be so abundant that oxygen doesn't get a chance to slip into the active site. So, excuse me, we looked at this. We say it's a spatial segregation. Boy, oh boy. And so we have two different physical, oh boy, excuse me. We have two different physical locations of where things are happening. So you see the mesophyll cell and the bundle sheath and the Calvin cycle. So most of the Calvin cycle is in the bundle sheath. So that's a spatial segregation. And then you can see that the C4 cycle actually kicks off in the mesophyll cell. So you have to imagine sunrise coming up. The plant is getting bathed in sunlight. They start to open their stomata and take in CO2 for the light reactions of photosynthesis. With C4 plants, they don't immediately react it with rubisco. Instead, they use PEP carboxylase, which is a really good enzyme because it's so picky. It only can take carbon dioxide into its active site. And so that way it's able to get the carbon dioxide essentially fixed into the malate molecule. And then the malate gets shunted, physically moved from mesophyll to bundle sheath. So it's kind of like a vector. It's delivering the carbon dioxide to where rubisco is located in the bundle sheath, right where the Calvin cycle is going to kick off. And then because this is a cycle, we see that the other product, pyruvate, once CO2 is cleaved off, that leaves you with pyruvate, and then that has to go back to the mesophyll. And you can see the plasmodesmata and the little tunnels between the cells are doing that transporting. And then the pyruvate will go into a reaction where ATP is expended. So this is not free, but with ATP's help, you'll end up with PEP. So basically, by the time this is all done, what you're seeing is that malate is delivering that CO2. And if you can imagine this happening over and over and over and over and over, you can imagine how CO2 is really starting to bathe or build up around rubisco. Okay, questions on the C4? So we worked through that. And we looked at how they have that special spatial segregation. Their anatomy is very unique. You can see under the microscope, you can get clues to what plants are straight C3 plants versus ones that are C4, because C4 plants really have that beefed up ring of cells around the vein. We call that the bundle sheath. They're very plump, enlarged, and they have a lot of chloroplasts. And then typically you see there's spongy mesophyll that kind of hug up to the bundle sheath with lots of plasmodesmata connections, those tunnels, so that you can get really good transport of the malate and the pyruvate back and forth. You have that cycle going on. Okay. So then we need to look at this one too. So this is another group of plants that evolved in very drought-prone, hot conditions. So now we're thinking desert environments. where we have really hot days, extremely hot. And then relatively cool. If you've ever camped in the desert, you know that the nights can get cool and a little bit more humid. So now CAM plants, they don't have a spatial segregation, they have a time or temporal segregation. So some things they do during the day and some things they do at night when they're running their cycles here. So they're trying to show this with this drawing, how at night, if you are, a giant saguaro cactus in Arizona, it's not the best idea to have your stomata open during the day. What will happen? Yeah. Oh my gosh, you'll dehydrate so quickly. And so that's not the best strategy. And so what they're going to do is keep their stomata closed during the day, and then they'll wait for the night when it gets cooler, much cooler, and a little bit more humid. Then they'll risk opening their stomata. However, they can't do photosynthesis at night because it's dark. So they can't do the light reactions. They can't even do the Calvin cycle at night. It won't run without light either, or the enzymes won't. So they got to store it up instead. So what they're going to do is they're going to take in that CO2 at night. As you can see, some of the same exact players are here as what we just looked at with the C4 cycle. So you still have that PEP carboxylase enzyme that's really good at only interacting with the CO2 and not O2. And then you'll get CO2 and PEP reacting, okay, with the help of that PEP carboxylase. And you'll get oxaloacetic acid again, OAA, which gets converted to malate, okay? And then what happens is instead of malate getting vectored anywhere, it gets stuffed into the vacuole of the same cell. All right, so this is all one cell, and they're showing that the malate gets stored in the vacuole all night long. You can follow this with a pH probe. You can put a tiny little pH probe into the vacuole, and you'll see the pH getting more and more acidic. The pH drops as malate, because malate is an acid. And so it will build up all night long. So as the plant's doing that, you're getting that malate in the vacuole. Then the sun comes up and they're like, oh, I'm going to close down my stomata so I don't die of dehydration. But now, because of the light, they can do the light reactions, they can do the Calvin cycle. So now they got to deliver the CO2 over to where it's needed. And so they're trying to show that malate will come out of the vacuole in the day, and that CO2 gets cleaved off of it and fed right to Rubisco in the chloroplast. So it's all one cell. It's not like we have a separate mesophyll and bundle sheath that are doing different things. It's not that. It's all one cell. It's just certain things happen at night, certain things happen during the day. All right, let's in words a little bit here. So that malate acid is going to be accumulating. So I believe this picture is from your book. And they're showing malic acid building up in the vacuole here all night long. And then during the day, malate comes out, the CO2 gets cleaved. And Rubisco's in the chloroplast there, and it's just getting fed a lot of CO2 through this action. Okay. So similar players, it's just a slightly different look. And if you think about it, I mean, if you can imagine, if you've ever been to the desert, well, boy, does it get hot during the day. I mean, it's pretty drastic when you have to use an oven hot pad to open up your car door. My nephew used to live in Arizona. My question is, if you live in Arizona and you're going grocery shopping, how do you not get your ice cream to melt on the way home? What do you think they do? Seriously, it's like, what do you do if you want ice cream? It's going to be a puddle by the time you get home. They must have special coolers they bring to the store and put it in the cooler right away. Or you just don't eat ice cream, which I can't imagine a life without ice cream. All right. Here are some cam plants that you may be familiar with. Hey, when you did your propagating, did you look at, there was at least one plant that had the little plantlets on the leaf edges, the mother of thousands, it's called. That's a pretty hardy plant. So you can even see in this image, the little roots coming off. So if you just pluck those off the mother plant and put them into soil, they'll grow pretty quickly into a brand new plant. It's a cloning mechanism that the mother plant has. So really cute. We used to have a lot of those in the greenhouse, and then, well, we had a modest amount, and then all of a sudden we had a huge amount of these plants because I found out why. My grad student at the time, he'd go in there to increase humidity. So he'd take the hose and he'd just spray across, the whole greenhouse. And of course, the jets of water would hit those. They'd fall off the plant and land in the soil pots all over the place. And pretty soon we had those plants everywhere. So I'm like, how did this plant get over here? And, so once I figured out how he was watering, it made sense. This one's a funky one. This is actually, believe it or not, a gymnosperm plant. I know that's really weird to think about. We're gonna learn about this one. Very odd plant, but it actually belongs into the gymnosperms. You can kind of see that it's forming cones here. So that's called welwitschia. And then of course pineapple. Pineapple is a cane plant, very important food crop. So lots of succulents, as you can imagine, because most succulents are native to the West habitats here in America. So the giant saguaro, beautiful, beautiful cactus. It's actually, the flower is the state flower. So when they were looking at plants that do this CAM cycle, they named it the Crassulaceae acid metabolism, which abbreviates down to CAM, because the first few plants they found belonged to this family, the Crassulaceae family. Unfortunately, they also figured out that lots of other families do that, but the name was already implemented and it kind of stuck. Apparently, botanists don't like to break with tradition, so they just kept the CAM cycle term. But you need to realize it's not just Crassulaceae family. There's many other families where you can find plants that do this. And then if you look at one family of plants, some can do CAM and some can't. In fact, you're going to find in families, that in one family of plants, you can have everything. You can have just C3 plants, you can have CAM plants, you can have C4 plants. So when they have been tracking this and studying it, they think the CAM pathway actually popped up 60 independent times at least. And that's why you have so many different families with these different cycles, lots of independent processes going on over the span of evolution. So these are some of the more important ones that are either food production or things that you're used to seeing, and lots of succulents are sold also for houseplants. They're pretty easy to take care of. Most of the time, the biggest issue I see with people who have succulents is they're too nice to their succulents and they overwater them, that they get waterlogged and die. So be very stingy. Be mean to your succulents. You have to withhold water a fair bit, especially in winter. Vanilla, actually, we get from an orchid. That's the only orchid that we see that is involved in some kind of food. What do we get from agave? Tequila, yeah. So very important, multi-million, if not billion. Whoops. And then the primitive plant, that is getting at the idea that it can show up in gymnosperms. So gymnosperms are more primitive compared to angiosperms. Okay, so you see the CAM showing up there. So the CAM plants have recently been popular in terms of research because of climate change issues. They're trying to figure out how plants can withstand drought and heat so well. And is it possible to create plants that are more CAM-like in terms of how they survive? And so they're really starting, there's a lot of scientists that are studying different CAM plants and looking for ways to use what they're learning as we are dealing with climate change. So this is something that you'll probably hear more about over the years. Really, for a long time, CAM plants were kind of ignored, unless you lived in the West where they were very common. They were rather kind of obscure in terms of research, but not anymore, not with climate change going on. So if you've been out west, beautiful cacti there. If you look at what's going on with, how does this improve their ability to handle drought conditions? They usually will assess it this way. They'll say, okay, per gram of carbon dioxide, what do we got going in terms of water loss? And if you're a CAM plant, it's only 50 to 100 grams of water loss. For C4, it jumps to up to 300 grams of water loss per gram of CO2. And of course, C3 loses the most water. And so this is just trying to show that if you are a desert plant and you're doing CAM, your ability to not dehydrate, you're pretty good at it. And so when you start thinking about these different cycles, if all plants can do the C3 cycle, and then you have these special plants that tack on the C4 or the CAM, depending on where they live. So now when you are traveling and going to these different areas, you can get an appreciation for why certain plants have adapted so well. And it's not like they've met with perfection. It's just they've adapted to their current environment. So, like corn, maize is a C4 plant. And there's a lot of farmers here in the Midwest area that grow corn. And that's not ideal in terms of because the C4 cycle costs extra energy and they don't really need to do it up here in the Midwest. The C4 plants don't really need to do the C4 cycle. And so in that respect, it's not ideal for the C4 plants, but you go back to where they were native to, where they evolved, and it suits them quite well. And of course, all your house plants or plants that you may landscape with, it's a good idea to know what type of plant you're working with. Because like I said, you want to be kind of stingy with your succulents. You don't want to overwater them. It actually does more harm than good. And so keep that in mind, thinking about how they do their Kelvin or their carbon fixation. And when they look at the process of how these cycles evolved, like I said, all plants do C3. So all plants started with that. But Rubisco being sloppy and not telling the difference between CO2 and O2, well then the CAM pathway evolved next. Certain plants in the desert really were desperate for some way to survive. So the CAM cycle came about and then the C4 cycle. So that's kind of the evolutionary process. Questions on these three cycles?
Julie, you just answered it when you said all plants do C3. Like, what are some plants that can do C3 and C4 or CAM and C3?
So if you think about like with corn, I'm not sure I'm understanding, but like with corn, it runs the C4 cycle and tacks it on to the C3 cycle. Is that what you're asking?
Yeah.
Which reminds me, so with the CAM cycle, there are, so far they've discovered a handful of plants that can turn the CAM cycle off. And so if you've lived in the West at all, which I haven't, but my nephews have, so I hear a lot about the weather out there. And you get the monsoons that sweep through. I don't know if they call it a monsoon out there, but there's another word for it. Hoodoo? No. Has anyone lived out west? It's a funny name anyways. So when the rain comes down, it's usually this deluge of just, and that's why they have the cities have to plan for it, so the water runoff goes into the channels rather than flooding the roads. Anyways, under those conditions, they're like, I've got plenty of water, so they turn off the cam. And then once all that water has gone away, which probably is fairly quickly, because it probably soaks into the soil pretty quickly. And then there's long periods of drought again. So then they switch back to the cam. And so they're looking at those plants too. That's a very active area of research too. How do those do it? So for me, when I have succulents as houseplants, and if I have them in my bedroom or any of the bedrooms, I purposely am really stingy with watering because I want them to do the cam. Why would it be, why do you think you'd want cam plants doing the cam cycle at night in your bedroom? No, not really. They're not doing photosynthesis per se, but instead of releasing, what are they taking up? So you're exhaling CO2 all night, plus a lot of other gases too in your breath, but um, they're taking in that CO2 all night long, which actually cleans up your air. It's good, improves air quality. And so if you do have succulents in your bedroom, just to hedge your bets, I don't have memorized who can turn off the cam cycle, but I make sure that my cam plants are not watered excessively. And if I have them in my bedroom, that's a good thing. So, okay. So we talked a lot about these cycles, but don't forget when they are doing this, one of their goals is to get their sugar made. Okay. So now we're looking at the chloroplast, where we have the light reactions of photosynthesis, the carbon fixation reactions. So that's the Calvin cycle. And then you see two yellow boxes, starch or sucrose. So what they have discovered is that the plant is constantly assessing what its needs are all day long, or, when it's running the Calvin cycle. Yeah, just during the day. And so they're like, okay, what should I do? Should I make sugars or should I pack it into starch for long-term storage? And so if the plant has plenty of sugar made, and that's not a problem right now, they're not short on sugar, they'll choose to keep the Calvin cycle running, and then that G3P as part of the Calvin cycle will be used to make a bunch of starch. And then they just pack away the starch in their chloroplast. It's kind of weird that they use their chloroplast for storage, but sometimes with a really well-fed plant, when you look at it under the microscope, you'll see a bunch of starch grains in the chloroplast. However, if the plant is, really actively growing and it needs like sucrose, right now I need it to be sent through the whole plant to different parts, then what they're going to do is they're going to take that G3P as part of the Calvin cycle and they're going to send it out of the chloroplast. And once it gets into the cytoplasm, it runs through all these reactions that you're seeing, and then you end up with sucrose getting made. All right, so that's why I was emphasizing last week, that the first sugar really isn't glucose that they're going for. That's not really what they want. They want to make sucrose because it's a very nice molecule for them to send through their whole plant body. It's well designed to be sent through their body to get where it needs to go unaltered and do its job of feeding the roots or maybe a baby leaf. Okay, so it's an either or though. The plant will tightly control this. I'm not going to go into details, but they tightly control what should I do? Make starch or should I make sucrose? And it's in each cell with each chloroplast, it's an either or decision. Okay. So it's the sucrose. If they want sugar, they're going to make the sucrose. And then that gets sent through the whole plant. We'll talk a little bit more about that later when we get into more of the anatomy of the plant. Okay. Questions on any of the cycles right now? please do that assignment. That assignment is designed to get you thinking the way you need to think for the exam coming up, okay? And it's easy points. If you sit down, and you can do it in groups if you need to, want to, whatever. This second exam, because it has these cycles on it, historically, this is a tougher exam. A lot of students, the average, class average kind of drops a little. So please, I'm not trying to scare you. I'm trying to prompt you to please study, right? Because if you don't have it into your long-term memory by the time the exam rolls around, you start to confuse the cycles. I've seen it time and again. So please, I'll do another assignment as well. We're going to do another assignment on C4 and CAM next week. All right. So I'm trying to do these assignments to get you prepped for the exam. But now I wanted to switch gears a little bit. Now I know on the syllabus, I think we're supposed to get into soil and water, but because of the snow days and cold days, we're a little bit behind, and I want to try to keep what we're doing in lecture kind of concurrent with what's going on in lab. In this week in lab, you're doing roots. So I want to get into the root lectures. And then what I'm hoping to do is the soil and water content will push to somewhere around the leaf. When we start covering leaves, we'll probably do that then. So I'm not trying to get rid of it. I just need to rearrange so we stay on track with what's happening in the lab. So if you are a root system, if you could look up, this is what your view would be. You'd see your shoots and leaves above you, in this case, tree trunks and beautiful tall trees. And so again, really odd body. I mean, can you imagine half of you being buried and half of you above ground, and very different habitats, different functions? So we're going to dive into the roots first, into the ground, and look at the roots. And we've had an intro to this already during week 1, but now we need to get into a little bit more detail. So we know, of course, that roots are definitely helping support the plant. It keeps them locked in place to where they germinated quite nicely. Here you're looking at the idea that they're taking up water, and they're really zooming in to the root hairs. So don't forget, it's really the root hairs, that are doing the lion's share of absorbing water. And they're not just absorbing pure water, it's also minerals in the soil that are dissolved in that water. So they're taking up water and minerals, and they need those minerals as part of their diet, just like we need our ration of minerals. We also have dissolved gases in the water to think about. We have that going on. So the same gases that are, here in the atmosphere, it's in the soil too, in the air pockets. You have oxygen and CO2 and nitrogen and all of that. It's, if you don't have a waterlogged soil, you definitely have nice air pockets in there. You tend to see this going on where they show you these two root systems. And I get why they do that. And it's showing you the extremes. So on one end, you can think of like the dandelion, which has a really deep taproot. If you've ever been given that job by your parents to go dig up dandelions out of the lawn, they actually make a special tool for it because the taproot is so deep that it helps if you have the special tool. I never had one, and I had to pull it out. But anyways, you can see that really long, thick taproot with much finer roots coming off. And then you have the other extreme to the taproot, which is fibrous. They call that fibrous root system. So that's the fibrous is like, pretty much every root is the same length, same diameter. They all look pretty similar. But I want you to appreciate that there's root systems in between those two extremes. Okay, so this is just kind of like bracketing. These are the two end extremes, and then there's several ideas of what can happen as a continuum from those two. By the way, Dandelions. You know, right, that dandelions are actually really good to have. You know about no mow May. You're not supposed to mow your lawn in May, so the dandelions can feed the bees. So anyways, I get very irritated with people who, well, it's not just people. Well, yeah, I mean, my brother lives in a neighborhood where the lawns have to be pristine, not a weed in the whole lawn. And I don't know. I don't understand why the war on dandelions. What is wrong with dandelions? They're so useful. Anyways, it's one of my pet peeves. Whoops, root structure. We'll get into this now. So I do need you to know the regions of the root as being shown here. And so this is a longitudinal root that's been sliced down the length. And we're looking at it. And so, of course, you have the root cap that we learned about previously. Keep those, keep the root tip and that apical root meristem tissue protected. And so you have your apical meristem right behind the root cap. Lots of active cell division. So anytime you think about meristem or meristematic tissue, that means really active growth. That's their job. Then if you look up a little bit, you'll see a region of elongation where physically those cells are getting bigger, longer. And then you'll enter into a zone called the region of maturation. And that's when you start to see the cells have reached their full size. They got everything up and running, and they're starting to make their root hairs. Okay, so the maturation zone is where when you finally see some root hairs showing up. So of course, this is a drawing. They're trying to also show here with all the different colors that the cells have differentiated to get into their designed function. So of course, some of it has to be vascular tissue, some of it's just parenchyma tissue, some of it's epidermal tissue. All right, so you can see in this lower region, they're not quite all there yet. Their functions aren't quite done. So they're still cooking. All right. Under the microscope of a real root, and you can see the root cap region here, and then the apical meristem. And then the elongation area, you don't even see the maturation zone here. Another view. So you'll be looking at root hairs today, or not today, this week in lab. So here, some of this terminology, don't worry about it. This is the elongation area where they know like which cells will develop into which tissue type. And so they've given them those names. Yes.
Do the root hairs just have like the anatomy of a normal root, but just on a smaller scale?
No, it's actually just an epidermal cell that has elongated. And so it's an epidermal cell. Yeah. And it's just really good because of that long, thin design. It's just its surface area is raised up quite a bit, and that helps with water absorption. Okay. So here you can see lots and lots of root hairs. What's the embryonic root called again? Radical. So that's a great image of that. Here's another one. So when they put the word pro, that just means it's not quite there yet. So cambium, procambium means it's going to become vascular tissue eventually. Protoderm just means eventually it'll become epidermis. So don't get too caught up on that. Okay, let's do a cross section of a root. So most of the root tissue is parenchyma cells, bunch of parenchyma cells that are used for storage. And they call that region the cortex. Okay, so the cortex is just the name they give for the region. So most of what you see in this image, that's all parenchyma cells. You can see the dark grains. That's actually like a dark purple. So those are starch grains. Those are amyloplasts that were stained in those parenchyma cells. So you can see that roots are really good at storing starch. That's another one of their main jobs. And then, of course, a young root will have a very thin epidermis, but definitely the bulk of what you're looking at is a bunch of parenchyma cells. And they say this is a dicot root. So when you think about angiosperms, the flowering plants, we're going to see some notable differences between a dicot and a monocot in terms of their anatomy as we work through roots, stems, and leaves. So they're blowing up now in this right picture. They took the central region of the root, which is the vascular tissue area, and blew it up. And so here you can see that the vascular tissue is, smack in the middle of the root. You can see that the bigger cells are xylem. Remember, anything that's stained red means there's a lot of lignin, which means it has a secondary cell wall, which means it's dead. Okay, so lignin popping red there with that fluoroglucinol stain. So the xylem, the vessels are the bigger ones, the tracheids are the smaller ones. You can see there's a handful of nice large vessels. And then usually when they do the stains, the phloem will be kind of a bluish color when you look at those. Phloem is living tissue, all right, they don't have a secondary cell wall. Then this red ring of cells surrounding all of that is called the endodermis. I need you to know that, please. So the endodermis is a ring of cells. And we're going to see that they have this these special strips that they named Casparian strips that are embedded in the cell wall. And they're very waxy Casparian strips. So suberin is a type of wax. I'll show you a better image of that in a minute. Basically, when they were like, this is kind of interesting. Why do we see these bands of suberin? And when they studied why the plant is doing this, in a nutshell, what it's really helping them do is making sure that water and the dissolved minerals keep going into the xylem from, bringing it from the soil through the root hairs and getting it through the root all the way to the xylem, and just keep it going in that direction, never having the water back out and flow out of the xylem. You want it all moving from the root hairs, soil, root hairs, all the way to the xylem, and then it'll get brought through the plant. So this is kind of the idea we're looking at here, where the black line is the water with the dissolved minerals. They're trying to show how the root hairs are so important for that. And then you can see how the water is traveling kind of like around the cells, not through the cells. They think that's the most efficient way of getting water moved to where it needs to go, which is the xylem. And then the Casparian strip, so the endodermis, they've blown up and they're showing it on the right. And you can see these bands, the red bands like that. That's the Casparian strip. So I think of like a UPS box, if you put a rubber band around it. It's kind of that idea. All right. And so that's really, really waxy. And in a nutshell, what it's creating is a way for water to go really only one way. It makes it get dumped into the xylem and it doesn't give it a chance to flow back out of the xylem. You want everything going into the xylem, all that lovely water. You don't want it to back out of the xylem. Get it in and then it'll start to course through the plant body. Questions on that idea? The other thing, right to the inside of the endodermis is this pericycle. It's another ring of cells, okay? And when you look at that area, it's an active area of growth. Okay. And it actually gives rise to lateral roots. It's kind of weird how the pericycle will grow and divide and produce more cells, more cells, more cells, and then they'll morph into a lateral root. You can see it's working its way out of that root, and eventually it's going to come out into the soil and be a lateral root. So you have the endodermis, and then to the inside of that is the pericycle. Here's a drawing with a little bit better labeling, I guess. Maybe a little bit easier to see. Okay. I think I'm going to stop there. I wanted to open up the exam and have you look through that. We will continue covering roots on Wednesday. So by the end of Wednesday, you should have what you need for lab.
Going back to the picture where it showed the Castorian strips, just a little confused. Like, are they only on the outside, or is it like the cell is here and then it's like a wall? So
It's like part of the cell wall, but it's not the entire cell wall. They just do a discrete buildup of suberin in a band-like formation in the cell wall.
how does that prevent water from coming back down? Is because if it's not on the inside and water's passing through, I don't know
Yeah.
how to explain what I'm thinking.
Yeah, you've jumped into plant phys there. And so basically what's happening is when the water is in the cell wall area, so as it's traveling here, it's in the cell wall area. So as it comes up here, it bumps it, all the water bumps into that wax and it can't go any further.
So
So then
forces
it
to
dumps
pass
it,
through.
yes, it dumps it into the cytoplasm. And that's what keeps it all one direction.
And
Mhm. All right, so if you get in, let me open it up so that you can